Good afternoon, ladies and gentlemen. I hope you can all hear me quite clearly at the back. Yes? Hello. This afternoon I'm going to go through some instances of characterization of biosimilar products. And what I want to talk about really, I can summarize in the why, what, and how. I've only got 25 minutes, so I can't go into much detail. So this really is a very quick overview of why, what, and how. Why is analytical characterization so important for biosimilar development? What are the regulatory issues that are associated with the characterization and testing of the biosimilars? And how can we characterize these types of products, bearing in mind that these are complex protein and glycoprotein products? So what package of analytical tools will be appropriate for these types of products? So before I start, just to define what I mean by biosimilar, because I'm sure you're probably all fully aware that there are many copy biological products out there. You can go onto the internet and find many of these for sale. The copy products may not have gone through a full testing against a comparator originator molecule. And so generally nowadays, these are termed non-comparable biologics, just to differentiate them from the biosimilars that have been going through the comparison versus the originator. And then we have another class of molecule that you're probably all aware of, the biobetters. And in the words of the famous song, this is something completely different. A biobetter, by definition, is not a biosimilar. However, all of the testing techniques that we utilize for biosimilars are equally applicable to biobetters. So bear that in mind while I'm talking. So if we look at, within the EU, a definition of a biosimilar, and this was from, sorry, we've gone slightly wrong here. Sorry about that. The biosimilars are molecules that have gone through the comparison, a head-to-head comparison with an already existing biological product that has come off patent. And this is the way that many regulatory authorities throughout the world are dealing with biosimilar products. So if we look at these regulatory authorities first, I think we'll start in Europe because we can quite rightly say that the Europeans were leading the pack in this development. Back in 2005, the EMA started with guidelines for biosimilar products. And they have really got a lot of experience in looking at this class of molecule, now with 14 different types of biosimilar that have been approved. So they're highly experienced. The rest of the world then followed, tried to catch up with Europe, and many other countries throughout the world have developed biosimilar guidelines. Some have adopted the EMA guidelines wholesale, others have modified or written their own guidelines. And there's been quite a lot of activity within 2012. Many new guidelines have been published, many draft guidelines have been finalized, and some countries, for example, India, in June, have come up with biosimilar guidelines as well. And also the WHO have guidelines which were published in 2009. Rather late to the party, we had the United States. And although we now have a legal pathway for biosimilar molecules in the United States, it wasn't until 2012, this year, in February, that the FDA published some guidance documents, draft guidance documents dealing with quality and scientific, and also Q&A, to try and explain fully these rather complicated set of legal documents. I mentioned that the European authorities had already approved 14 biosimilars. This is the list. What you will notice from this list is that they're all relatively small protein molecules. Epitin, filgastrum. Epoetin, yes, is large, but that's because more than 50% of its mass is due to carbohydrate. But they're quite small. What do we have coming along, though? We have in the pipeline insulins and interferons. In fact, as we speak today, there are 3 insulin applications sitting with EMA. But there's also the potential for monoclonal antibodies because the patents for these classes of products are starting to expire in 2014. And these are really the blockbuster drugs where the end result, the investment that you put in, might reap rewards in developing a biosimilar. There are guidelines for monoclonal antibodies, and they do throw up some particular challenges for analytical characterization. So they're not as simple as those simple glycoproteins such as human growth hormone, erythropoietin, etc. There are also issues of immunogenicity, which again have to be addressed. But as I said, there's great potential for these antibody biosimilars. And in fact, the first application for a monoclonal antibody went into the EMA earlier this year. That is for a monoclonal of infliximab. And the applicant is from a South Korean company based in Incheon. And they have, in fact, already received authorization from the Korean FDA during the summer for marketing of this product. So we wait to find out what the situation will be with EMA. But even more recently, there has been a second applicant to EMA with the same infliximab product. So why do we have to characterize? Why is it important to characterize? Well, with the biosimilars, characterization forms the first step in any analytical development pipeline. The comparability approach has to be carried out at the 3 levels: quality, safety, and efficacy. And the biosimilar has to be compared to a reference product. The objective is to determine if the proteins, glycoproteins, have the same biochemical and physiological properties, and also investigate the product-related variants and impurities. And it's not just the primary protein sequence that has to be the same. The secondary and higher order structure should also be compared, and particular attention should be paid to the post-translational modifications. So I mentioned that this could be carried out in a stepwise fashion. So stage 1 would actually not be looking at the biosimilar molecule, but a step beforehand, the thorough characterization of the originator molecule. This is to determine really what is now called the quality target product profile, to find out what the target is for the biosimilar to be manufactured to. And this will invariably mean that some de novo MSMS sequencing to determine the exact protein sequence is required, because that exact sequence and all its post-translational modifications will not be easily discovered from literature searches, and you're certainly not going to get the originator telling you what that structure is. So you have to do all the hard work yourself. And it does employ some very exacting analytical tools, such as MSMS de novo sequencing. And I will show you why this is important in a few minutes. So that's stage 1, determining the quality target profile, not just on one batch of biosimilar, but one batch of originator, but looking at the range. So to get some idea of the specification range of the originator molecule, which will change over time. Second stage then is the comparison side by side. And as I mentioned, primary and higher order structure, and then look at the biological activity, look at the bioassays, the cell-based assays, look at the virology, etc. So a lot to do at stage 2. That really is the meat of the development program. On to stage 3 then, the clinical comparison and the bioanalytical. But not forgetting that throughout all of this, you can think of it at stage 4, but I prefer to think about it as spread over stages 1 to 4. The formulation, that will require characterization. Quality control assays and stability assays, all of these will involve analytical characterization at some stage. So I mentioned why it's very important to ensure that you are shooting for the right target. And this is a case study that we have experienced at SGS MScan, where an innovator product was being examined for its structure. And we were unable to find from the written structure a section of the protein. There was a peptide of the protein that we couldn't confirm. Now, for the people amongst you that think that we might have designed a new amino acid called X, the X's are just there to protect client confidentiality. But what we couldn't find was this tryptic peptide. We still couldn't find it when we searched for the missing tryptic cleavage point here. So we performed some MSMS analysis, and to cut a very long story short, we could find a signal that from our MSMS spectra, we were able to deduce some sequence information for the anticipated peptide. However, the peptide was 10 mass units larger than we were expecting. And after a lot of confirmatory MSMS experiments, we were able to deduce that at the residue number 2, where this 10 mass unit difference was attributed, there was a change from a serine to a proline. So from 87 mass to 97 mass, hence the 10 mass unit difference for the molecular weight. Now, this just shows how careful you must be in interrogating that initial originator sequence so that you can be absolutely confident when you go on to design your own biosimilar. So stage 2, we're then going on to look at the comparison side by side. That throws up, again, many challenges. For example, the choice of the reference material, and we could talk for hours about that issue. The issue of making sure that you look at multiple batches of the biosimilar versus multiple batches of the originator, because I'm sure you're aware of recent publications that shows the changes of originator products, quite legitimate changes, because these will have been performed under ICH Q5E as comparability changes, but they will change the product over time. So now let's look at the question, what combination of analytical techniques to look at the primary and higher order structure might be useful? Again, it's an open question. There is no one technique that will do everything, and there are no set rules. It's very much matching the techniques to the anticipated problems, post-translational modifications, etc., that you might find within your product. But one very good guideline, signpost, menu, or some people can also call it a Bible to follow, is the ICH Q-Topic Q6B, and it gives a nice list of 6 structural characterization areas to examine and 6 physicochemical properties to examine. So in one page you have a very nice menu, but it might not be necessary to do everything for your particular molecule, or it might be important to do more than what they recommend. So it all depends on your molecule. Another source of information on what to do, if you look at the EMEA, or the EMA as it's called now, their revised monoclonal antibody guideline. Now this is the monoclonal antibody guideline that is looking at the original monoclonals, not the biosimilar one, and that has a lot of information about the types of techniques that are recommended. So if we put it all together in one snapshot, here is a typical analytical case study. A monoclonal antibody, and we're performing a variety of techniques, heavily using mass spectrometry. So we use mass spectrometry to look at the intact protein and the subunits. We do N-terminal gas phase sequencing. We do peptide mapping. And with a molecule of this size, 150,000 Daltons, we wouldn't get full sequence coverage unless we perform 2 or 3 different proteolytic digests. So that's important. But we aim for 100, and we do get 100% sequence coverage. And then there's a carbohydrate to think about. Obviously, single N-linked site with the antibodies, but other molecules, erythropoietin, the carbohydrate really is a much more complex carbohydrate. Plus techniques that will look at the secondary and tertiary structure, things like circular dichroism, and perhaps problem-solving techniques. Here we have analytical ultracentrifugation, and this is to look at aggregation of the molecule. So again, you tailor your toolbox of techniques to the particular molecule. So if we go down through the strategy, the first thing I mentioned was intact mass measurement. And with modern mass spectrometers, it's now quite easy to get good full resolution at 150,000 Daltons to be able to separate out the different glycoforms of a molecule at 150,000 Daltons. So these signals that are represented here are all differing by roughly 162 mass units due to the different carbohydrates, the sugar units that are on that N-linked carbohydrate. So mass spectrometry, very powerful. This is another example of mass spectrometry, and as I always say, those within the audience that can remember life before Microsoft Office will recognize this as an old-fashioned slide. That's because this is actually a historical example. It's the first published example of mass spectrometric peptide mapping of a biotech product, and it was published by MScan and Imperial College back in 1982. But this technique now has become the routine workhorse. Everyone performs peptide mapping. You can also couple the peptide mapping for larger molecules with online LCMS. And you can then generate routine peptide maps in a much higher throughput than before. With mass spectrometry, we can also, as well as detecting the molecular weights, we can also sequence or obtain sequence information. This can be done in a variety of ways, but one of the advantages with mass spectrometry is the ability to sequence from both the amino and the carboxy terminus. So the information can be confirmatory. However, mass spectrometry is not the only thing that we utilize in our daily work. We also use N-terminal gas phase sequencing, the classical Edman degradation. Why do we do this? Well, there's one particular reason. Yes, we can use it in the automated fashion to get, say, 10 or 15 amino acids in from the N-terminus. But in one particular case, it does have a clear advantage over the mass spectrometry method, and that's where we're dealing with isobaric amino acids. So in the case of leucine and isoleucine, and I'm sorry that this slide is very faint, but we're able to use the Edman separation technique using chromatography to be able to differentiate between these two specific amino acids. And again, it means that you always have to mix and match your techniques to the problem that you're addressing. Disulfide bridge analysis, another quite complicated, taxing, challenging thing to do. We can extend our peptide mapping strategy using the digests to obtain a mixture of peptides. The mixture of peptides, if we run under non-reducing conditions, the disulfide bridge will remain intact, giving a signal in the spectrum. If we then reduce the mixture, put the mixture back into the mass spectrometer, run the map again, and compare to the original map, the disulfide bridge will be reduced, it will break down, and so that single peak will disappear, but it will give rise to two new signals corresponding to the molecular weight of the two separated peptides with the free thiols. So if we look at a real example, some real data, here we have disulfide bridged tryptic peptides from our tryptic LCMS map. We reduce them, that signal disappears, and two new signals have arisen in our spectrum. Another challenging area, analysis of glycosylation. Now, there are many things that you have to think about with glycosylation. You have to think about the glycosylation site, both N-linked and O-linked. Then at that site, you have to think about the overall population of glycans, what the major carbohydrate sugars are. Then think about the individual monosaccharide units that make up those glycans and how they're linked together. So we have many strategies to address this, but it all starts, again, we use liquid chromatography methods and mass spectrometry methods. So if we think here in this simple cartoon, we have the N and O-linked glycans. We use the peptide mapping approach by digesting, juicing carboxymethylating to open the disulfides, digesting the specific protease, then using an enzyme that will remove the N-linked, for example, so you get a mixture of free N-linked and the O-linked and other peptides, separate those out, and then we could use a chemical method, perhaps a reductive beta elimination, to take off the O-linked and get a pool of O-linked. And these then can all be separated out and studied. So glycomics and the analysis of the carbohydrates we could talk for hours about, but basically multiple approaches, and it's important not just to look at the overall pool, but to identify the individual carbohydrates at each glycosylation site. So I've talked about some fairly sophisticated instrumentation, the use of LC-MS and MSMS, but not all instrumentation needs to be that sophisticated. We can use routine, simple, analytical quality control type instrumentation also in our comparability studies. For example, capillary isoelectric focusing, and here we have an example with 3 monoclonal antibodies, 3 batches. And then we can also look at the higher order structure. This is what ICHQ6B says. We need to use a technique such as CD or NMR. So again, variety of techniques, DSC, FTIR, etc., can be used. But there we're looking at a comparative manner. In this example, we have a CD analysis in the far UV, where we're looking at 2 batches of originator and 3 batches of biosimilar, and you can see that we can run those together, superimpose the individual data, and then we can compare them. We can also look at the near UV CD, and that will give us some indication as to how the tertiary structure of the molecule may be breaking down, for example, under stability. So again, we can use this in a comparative sense. So to wrap up the conclusions in this very quick overview, a snapshot of what we do with some of the techniques we have available within SGS. We use mass spectrometry really as our principal tool because of its strength in looking at the post-translational modifications, including the non-protein modifications. We can use these techniques at all stages, but we use them with the biosimilars principally in, first of all, obtaining the originator sequence, and then in the comparative sense. But MS techniques are not alone, and we use full physicochemical characterization tests in our toolbox to look at a biosimilar. Coupled with then, following the work that SGS, MScan have provided, we are able to provide bioanalytical, cell-based assays, and clinical services. SGS are then able to put together a full development package for biosimilar manufacturers. And if anyone would like to ask us any questions about this, we have a brochure here that is available on our stand over there on the left, and we'd be very happy to discuss our full analytical portfolio with you for biosimilars, biobetters, or any other biological molecule that you might have. So with that, I'd like to thank you very much for your attention. Thank you.